Hello everyone, today we will discuss about the biochemistry of the amino acids. Now first of all, we all know that all proteins are the polymer of amino acids which are linked together by the peptide bonds. Now in the nature we have around 300 amino acids, out of which only 20 are present in the human being body. All amino acids are alpha amino acids. What are alpha amino acids? As you can see in this picture that this amino group is attached to the same carbon atom to which this COH group or the carboxyl group is attached. So these type of amino acids are known as the alpha amino acids. So all amino acids are alpha amino acids in the human being body except the proline. Just remember, proline is not an alpha amino acid. Okay, now the classification of the amino acid, it will be done on the basis of four main categories. That is the structure of the amino acid, side chain, metabolism and the nutritional value. Now first of all based on structure, in that first of all aliphatic amino acid. Aliphatic amino acid, that means the amino acid won't be having any kind of cyclic structure. There will be only straight carbon line. Okay, in this we have three subtypes, that is the monoamino monocarboxylic, monoamino dicarboxylic and dibasic monocarboxylic. Monoamino monocarboxylic, that is one amino group and one carboxylic group is attached to the carbon atom. Okay, now in this we have further categories, that is the hydroxy containing, amide containing, branched amino acid, simple amino acid and sulfur containing amino acid. The example of these are first of all hydroxy, serine and threonine. Serine and threonine have OS group in that. Amide, asparagine and glutamine. Branched amino acid, valine, leucine and isoleucine. Simple amino acid, glycine, alanine and sulfur containing amino acid which will be having sulfur in it will be cysteine and methionine. So these all examples are very important. Now next is the monoamino dicarboxylic. That means the carbon atom will have one amino group and two carboxylic or the COH groups. The example of course this have two carboxylic groups, so of course it will be acid. So two things, aspartic acid and the glutamic acid. And the dibasic monocarboxylic, that means two amino group and one carboxylic group. So examples are lysine and arginine. These are some of the examples. Now the aromatic amino acid. Now in this amino acid, as the name says aromatic, that means they will be having aromatic ring in this. The example are phenylalanine and the tyrosine. Now the heterocyclic. In heterocyclic amino acid, mainly the carbon atom is having two different elements, and except these two elements, they would be having one amino group as well, nitrogen containing group as well. So examples are the tryptophan and the histidine. Amino acid. Amino, that means the carbon and the nitrogen compound of this amino acid, they will be attached by one double bond. So means C double bond N compound will be present in this type of amino acids. So example is proline. Proline is also not an alpha amino acid. Now next is the derived amino acid. Derived amino acid are further of three types, that is derived amino acid found in protein, derived amino acid not found in protein and the non-alpha amino acid. Now derived amino acids are first of all, after synthesis of the protein, some of the amino acids are modified, which means when the proteins are actually formed, some of the amino acids are modified. So derived amino acids are found in the protein. For example, hydroxyproline and hydroxylysine. Means in this, the protein is fully formed, but after that some derivation or some modification is done in that. And this hydroxyproline, hydroxylysine are very important component of the collagen. Collagen is a protein. Gamma carboxylation of the glutamic acid residues of the protein is important for clotting process. And next is the ribosomal protein and the histone are also modified, which are actually methylated and acetylated after their formation. Now the derived amino acid not found in the protein. Some derived amino acids are seen free in the cells. Okay, and these are these only. For example, ornithine, citrulline, homocysteine. These are mainly produced during the metabolism of the amino acid and mainly in the urea cycle. Thyroxine may be considered as derived from the tyrosine but not found in protein. And next is the non-alpha amino acid. Gamma aminobutyric acid or the GABA is derived from the glutamic acid. Beta alanine, where amino acid or the amino group is in beta position, and that this beta alanine is very important constituent of the pantothenic acid. Pantothenic acid means the vitamin B5 and coenzyme A. Now based on side chain, now these are mainly of three types: non-polar side chain, and the next two are the polar. But in this one is non-ionic, that means it won't be having any of ions, and next is the ionic. Non-polar side chain examples: alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine and tryptophan. Now in this, as because they are non-polar, they would be hydrophobic and lipophilic. Next is the non-ionic polar side chain. Example: glycine, serine, threonine, cysteine, tyrosine, glutamine and asparagine. Now in this, as because they are polar, so of course they would be hydrophilic. But the ions, but there would be kind of ions or charges in it. Okay. Now next is the ionic polar side chain. That means they would be having charges in it. For example, aspartic acid and glutamic acid. Okay. In this there are two further. That means acidic and basic because they are having ions. So in acidic, aspartic acid, glutamic acid, and basic, lysine, asparagine, and histidine. They of course they would be hydrophilic and lipophobic. Now next is based on metabolism. Now all these things, all these three, purely ketogenic, and glucogenic, and purely glucogenic. In this, it means that when this amino acid actually goes in our metabolic pathway, they would be converted purely into either ketogenic and glucogenic, or either purely glucogenic. So these are further divided into three parts. Okay. Now first of all, purely ketogenic, that means in the metabolic pathway, the amino acid will be converted purely into ketogenic or ketones or the hidden bodies. For example, leucine, ketogenic and glucogenic, that means in the metabolic pathway, the amino acid will be converted into ketone bodies or in the glucogenic. Okay, for example, tryptophan, tyrosine, lysine, isoleucine and the phenylalanine. Okay. Now next is the purely glucogenic, means in the pathway, in the metabolic pathway, they will be converted, they are purely ketogenic, means they will be metabolized purely ketogenically. All the remaining 14 means we have total 20 amino acid in our body. So we got all the 6 over here in the above one, in the rest 2. So in the rest, purely glucogenic, all the remaining 14 will be considered. Now based on nutritional value, in this there are 3: essential, partly essential and non-essential. Now the essential one or indispensable are very important for our body because they are not produced by our body. So we have to take this essential or indispensable from outside. There will be supplements of this amino acid from outside because they are not produced in our body. For example, phenylalanine, valine, threonine, tryptophan, isoleucine and methionine. Just remember mnemonics: PVT-TIM, private TIM. PVT-TIM. Now the partially essential, histidine and arginine, means these are partly formed and not formed in the body, means we have to take it as well, and if we are taking then it's okay, but if we are not taking it in our body, then there will be deficiency of these two. Next is the non-essential, means they are not essential for in the supplements because they are totally formed in our body, so there is no need to take this in our diet. Now all remaining 10. Now the properties of amino acid. Now first of all, the taste of amino acid: sweet, tasteless, bitter and artificial sweetener. Now sweet are the glycine, alanine, valine, serine, tryptophan, histidine and proline. These are the sweet one, means they will taste sweet. Tasteless are the leucine, bitter are the arginine and isoleucine. Now artificial sweetener, whatever artificial sweetener we add in our recipes, these are the made up of aspartame amino acid. Now ampholytic and isoelectric point. Now first of all, before this, I would like to tell you that all amino acids have high melting point, that means more than 200 degrees Celsius. All amino acids are soluble in water and alcohol. That means polar solvents, means soluble in water and alcohol as well. But they are insoluble in non-polar solvents, that is the benzene. Okay, now the amphilytic and isoelectric point. Now the pH at which the molecule carries no net charge is known as the isoelectric point. Very important line. Now in acidic solution, they are cationic in form, and in alkaline solution they behave as, of course, anionic. Means in acidic they act as positive charge, and in the negative they act as negative charge. So whatever, because they are isoelectric, because their net charge is 0, so in whatever medium they are added, they will act like that. Now at isoelectric point, the amino acid will carry no net charge. All the groups are ionized, but the charges will cancel each other. Therefore, at isoelectric point, there is no mobility in any electric field. Isoelectric point has plus and minus charge both. Minus charge by the COH, positive charge by amino group. So of course, there will be no mobility in any electric field at the isoelectric point. Now the solubility and the buffering capacity will be minimum at the isoelectric pH. Okay, solubility and buffering capacity will be minimum, while the mobility is also negligible. Okay, now the optical activity. Amino acid have asymmetrical carbon atoms, so that's why they show optical activity, except the glycine. Glycine don't have asymmetrical carbon atoms, so except glycine, all other amino acids show optical activity. Now, of course, you have studied that optical activity showing compounds have D and N configuration. So, D and L configuration, out of this, the L is found in the nature. So, L compound or the L amino acids occur in the nature, and therefore they are known as the natural amino acid. While D amino acids are seen in small amount in the microorganism and in the antibiotics as well. For example, gramacidin S, polymyxin, actinomycin D, and valinomycin, and as well as in the bacterial cell wall. So, where gramacidin S, actinomycin, valinomycin, and the polymyxin. Now, the isoleucine and the threonine have two optically active centers, and therefore each has 4 diastereoisomers. Okay, this is not that much important, but the main thing is that the glycine is optically inactive. Now, Sorensen formal titration. Now, amino acid cannot be exactly titrated. For example, for this, if I take 1 ml of glycine amino acid and 1 normal of NaOH, that means the sodium hydroxide base. So, if I titrate these two things together, so what will happen? The COH group of the amino acid, okay, will give its hydrogen compound to the hydrogen atom to the NaOH to make it to titrate it, right? Because NaOH is base. So to titrate it, the COH group will give its hydrogen to the NaOH. But we have NH3 or the amino group in the amino acid. Okay, so of course it is also base. So it will take the hydrogen of the COH compound and will not, and the alkali requirement will be less than 1 ml. Okay, because 1 ml we added the glycine. So of course the requirement of the alkali will be equal to 1 ml, but here the alkali requirement will be less than 1 ml. So this says that the COH group's H is taken some partly by the amino group of that same amino acid, and the requirement of alkali is decreased than 1 ml. So just in order to inhibit this thing, by the Sorensen formal titration, we will add excess of formaldehyde. So this will convert the basic or the alkali amino group of the amino acid into dimethylol derivatives, into neutral dimethylol derivative. So this is the Sorensen formal titration. This can be asked in the viva, that what is Sorensen formal titration. In this, the alkali requirement for the titration is less as compared to whatever amount we added, because the COH group is taken partly by the amino group. Now next are general reactions of the amino acid, very important for long question answers. Now first of all, we all know that we have side chains. That means the carbon atom is attached to the carboxyl group also, due to amino group, and due to the side chain of the R group. Now first of all, we talk about due to the general reactions mainly occur due to the carboxyl group. Now two things can occur over here, either decarboxylation or the amide formation. Now what is decarboxylation? Either we can remove the carboxyl group or the CO2 group from the amino acid. So what will happen if we will remove the CO2 from the histidine, so they will form histamine. Means over here amine group is formed. Just remember, due to removal of the carboxyl group from the amino acid, amine group is formed. And when we will add amino group, so amide will be formed. Okay, so examples are, first of all, decarboxylation. Histidine is converted into histamine and CO2. Tyrosine is converted to tyramine plus CO2. Tryptophan to tryptamine plus CO2. Lysine from lysine to cadaverine plus CO2. Cadaverine is the amine name of the lysine, and the glutamic acid into GABA plus CO2. GABA, that means the gamma aminobutyric acid. I told you before, the GABA is mainly used for the clotting process. Okay. Now the amide formation, in this the acids, monomer dicarboxylic compounds are mainly used. Aspartic acid, glutamic acid, when amino group is added, they will form amide formation, that is the aspartate and the glutamine. Now due to the amino group. Now in this, first of all, transamination. As the name says, transamination, that means there will be transportation of the amino of the amino acid. I have shown the figure, this figure. Over here, as you can see, this glutamate is having one amino group, and this alpha keto acid, let's consider this as a pyruvate. Okay, suppose this is a pyruvate alpha keto acid, so it is having one ketone group. So there will be transfer of this NH3 to this alpha keto acid and transfer of this ketone group to this amino acid. So this will form alpha ketoglutarate and alpha amino acid, or we can say alanine. Over here there will be alanine will be formed. So this, in this reaction, the NH3 is transferred to the other compound, and the ketone group of that particular compound transferred to the amino acid plate, amino group of that particular acid. So different amino acid will be formed, and so this process is mainly a very important reaction in the body for the interconversion of the amino acid and for synthesis of the non-essential amino acid. Now the oxidative deamination, as the name says, there will be deamination of the or the removal of the amino group, this amino group from the amino acid. Now the alpha amino group is removed from the amino acid to form the corresponding keto acid and ammonia. This is keto acid. Okay, so you can consider as pyruvate as well. Pyruvate is a keto acid. So keto acid and ammonia. Okay. Now in the body, glutamic acid is the most common amino acid to undergo the oxidative deamination. Okay, now next term is the trans-deamination. As the name says, there will be transamination plus deamination. So what happens is in this is that any of the amino acid will give its ammonia to the alpha ketoglutarate acid and will form the glutamic acid. Now the glutamic acid which is formed will undergo oxidative deamination to release the ammonia. So over here two reactions are metabolically coupled and often known as the trans-deamination. This one. This trans-deamination is a mixture of a transamination and the oxidative deamination. Now formation of carbamino compound, this mainly occurs in our body when we need to transport the carbon dioxide. Now carbon dioxide add to the alpha amino group. This is the alpha amino group, so carbon dioxide is added to form the carbamino compound. The reaction mainly occurs at the alkaline pH and serves as a mechanism for transport of carbon dioxide from the tissue to the lungs by hemoglobin. This is the carbamino hemoglobin. Now last is due to the side chain. Now first of all, we have transmethylation. What is transmethylation? The methyl group or the CH3 group of methionine over here. This is the methionine. So the methyl group, the CH3 group of the methionine, after activation may be transferred to an acceptor, okay, which becomes methylated. So methionine plus acceptor, so methylated acceptor, and the rest one is the homocysteine. Now the ester formation by the OH group. Okay, the OH group in the serine and the threonine. Okay, these two are the hydroxyl containing amino acid. So these two will form an ester bond. So the hydroxy amino acid can form the ester bond with the phosphoric acids. Okay, and in this manner, the serine and the threonine residue of the protein are involved in the formation of phosphoprotein. So when the OH amino acid actually forms ester of the phosphoric acid, they will form phosphoprotein. Okay. Similarly, if the OH compound will form O-glycosidic bond with the carbohydrate, they will form glycoprotein. Next, the reaction of amide group. Okay, amide group. Amide group are the glutamine and asparagine. Okay. Now, the amide group with the carbohydrate will form N-glycosidic bond and will form the glycoprotein. Okay. Now, over here, the hydroxyl group, the hydroxy amino acid, they are forming the O-glycosidic bond with the carbohydrate and forming the this glycoprotein. While the reaction of the amide group with carbohydrate with them, they will form the N-glycosidic bond and they will form glycoprotein. So just remember these two differences: O-glycosidic bond OH and N-glycosidic bond amide group. Now the last is the reaction of the SS group. What are the SS group? They are the sulfur containing groups. So now the cysteine has SS groups in it. So two cysteine molecules will be connected to each other by disulfide bond and they will form this cysteine. Just remember the difference between these two spellings. Don't confuse between these two. These are not having any E over here, as you can see. So due to side chain, we have transmethylation, ester formation by OH group, reaction of the amide group and the reaction of the SS group. So this was all about the biochemistry of the amino acid. We will talk about the protein biochemistry in another video. So hope you understood it well. Thank you so much.